So the EPIC-Round 2 project was born in the middle of the crisis of the SARS-CoV-2. The European Commission wanted to find a solution in Europe to find a treatment against all the possible variants of concern. The polyclonal antibodies of Fabentech could really answer to this question because of the polyclonality of our technology, and we can still prove that the product is very efficient against all the last variants of concern. So with the group that we have in Europe, in Germany, in Spain, in Greece and in France, we have all the expertise to answer to this very difficult question of the moving pathology. The patients are not the same, the variants are not the same, and so we need to find a solution for those patients who require desperately this treatment.
The main challenge is just the speed that the problem is just evolving. In our case, it's just the continuous and the speed of the evolution of the virus. So we have to constantly be monitoring all the changes that the virus is just experience. So we have to detect clinical isolates. We have to sequence those particular isolates, and we have to test the potency of neutralization of the tool that has been developed by the consortium, that is the polyclonal antibodies.
The biggest challenge is to have a model that can really reproduce the disease that is observed in patients, and in which we can test the therapies that are the aim of this project. There is still no therapy against SARS-CoV-2 infection, so we have a very effective vaccines that protect against severe disease for the general population, but still there is a part of the population who can suffer severe disease, so it will be very important to develop or to improve these therapies for this kind of patients.
This project is very important because it's a completely different approach. At the very beginning, there was the development of monoclonal antibodies, but it's been very disappointing because these monoclonal, they just are antibodies that just are targeting single epitopes, and the virus is being very fast changing all these epitopes so that they are escaping the immune response. The polyclonal approach that is using this project within this consortium is a different approach and has the advantage that the antibodies that are generated in the immunized horses, they are just targeting a high number of epitopes within the spike protein of SARS-CoV-2, which is the main component for viral entry, and we think that this is a good strategy to cope with all this variability and all this escape property of SARS-CoV-2.
The biggest challenge so far in EPIC-Round 2 is the constant change of the epidemiology of COVID-19, which is introducing us to frequent changes of the protocol design literature. However, the big offer coming from EPIC-Round 2 is that evidence so far suggests that SARS-CoV-2 is a novel pathogen which will remain for long as a pathogen in our society. So EPIC-Round 2 offers a completely novel treatment, a combination of immunoglobulins, what we call polyclonal immunoglobulin preparation, in order to stop the novel coronavirus.
As a conclusion, I would like to thank all the members of this consortium. They have brought to the consortium a lot of expertise and assets. We can work together much better, and we could prove that we can adjust ourselves to a very difficult and moving pathology as SARS-CoV-2. And this will be, I think, the basis of future cooperation we may have for other biothreats that will come. I know that the European Union is very sensitive about all these questions, and we could contribute to this preparedness plan that ERA is also very interested in.